Welcome back, everyone. It is my pleasure to introduce keynote speaker David Benjamin. David is the founder of the Living Architecture firm and associate professor at Columbia Graduate School of Architecture, Planning and Preservation. He's most recently the author of Now We See Now. David has been working at the intersection of biology, computation, and design for over a decade, and his work has been a huge inspiration for BDC since over a decade ago when we started GenSpace. Thank you, David, for joining us. We're really, really pleased to have you speaking this year. Thanks so much. David, you're muted.
Apologies. Thank you so much, Dan. It's really great to be here. Thanks for the generous introduction. And I remember back to those first days when I took a class of students to GenSpace, and it's really amazing what has happened since then. So I want to say that it's really great to be here, and I really admire what all of you are doing. I'm super happy to be able to share a little bit of our latest thinking and some of our recent projects with you this afternoon. But first of all, I'd like to acknowledge the incredible team that I work with. All of our work is very collaborative, and these people shown here individually and collectively bring it to life. As Dan mentioned, I'm a professor of architecture and a practicing architect. Here's one of our recent completed projects. And I like to start by saying that I believe buildings are living organisms. They breathe and pulse. They inhabit complex ecosystems of species, technologies, and culture. And understanding buildings requires understanding these vital signs and ecosystems. This kind of thinking allows us at our firm to expand the definition of environmental sustainability, and it also allows us to expand our design ecosystems as architects. So along these lines, I'd like to describe our design ecosystem at The Living, my design firm, which includes a variety of interconnected forces and involves a kind of hybrid of the familiar and the new, the ancient and the cutting edge, the technical and the social, the tangible and the atmospheric, the practical and the critical. So it's within this design ecosystem that I'd like to talk about a couple of examples of biodesign and our thinking about how biology can play into a broader design ecosystem. So we think of our design ecosystem as a kind of network of interconnected loops. And within this network, the topic of our work often addresses climate change, and our approach, as Dan mentioned, often involves new possibilities at what we think of as the intersection of biology, computation, and design. Of course, as you know, you're the group that would know this best, architects and designers have been inspired by biology for hundreds of years. But I believe, as you probably also know and believe, that biology of today is different. It's now possible to grow a cell alone on a glass slide instead of inside a living organism. It's now possible to visualize neurons firing inside of an animal in real time. It's even possible to cut and paste DNA and bring creatures to life that never existed before, such as yeast that produces an anti-malaria medicine. And as of just a few years ago, with the demonstration of CRISPR and gene drives, it's now possible to redesign or even eliminate an entire species very quickly, essentially molding evolution itself. In addition, it's now possible to apply some of the latest techniques of computation, such as computer vision and machine learning, to processes of biological growth. Biological functions involve hundreds of dimensions, but if they can be simulated in a computer model, then they can become a more actionable part of the design process. Yet, the more you learn about biology, at least for me, the more you see how complex biology is and how much we still have to learn about it. And in this sense, I believe that design with biology may involve design with a black box, design with forces beyond our complete control, and design with uncertainty. This new version of biology calls for a new method of design, a new approach. And in the past few years, we've developed 3 specific ways of thinking about this, 3 specific approaches to biodesign, and 3 categories for projects. And I'd like to describe them briefly here. The first one is biocomputing, which I define as using actual living organisms to process information and solve problems such as perhaps reducing carbon emissions. In an example of this approach, our firm has been working with Airbus, the big airplane manufacturer, to try to rethink the way that airplanes are designed and manufactured. But like many of our projects, we start off with a kind of tangible prototype. In this case, a partition, which is shown in red here, and it's the dividing wall between the seating area and the galley of a plane. And like many components in an airplane, this one has to be certified through pretty intensive testing. Here you can see the so-called 16G crash test of one of the partitions that's flying in today's A320 planes. And you get a sense of how much force this object needs to withstand in a potential impact of an airplane. And although this component looks a little boring, it's kind of forgettable, you've probably seen it several times on a plane and not thought much of it, it's actually a very difficult component to design with traditional engineering approaches, especially if you want to design a lightweight component. And Airbus, in reimagining the future of flight, was thinking about how it could reduce the weight of airplanes, which would reduce fuel consumption and therefore carbon emissions. So we started off trying to design a partition in a lightweight way. It can only be 1 inch thick. It can only attach to the fuselage, the airplane's main structure, at 2 points above and 2 points below. It has to hold the fold-down cabin attendant seat. And it's a difficult component to design in a lightweight way with traditional approaches. And this is where we turned to a living organism, in this case, Physarum, also known as slime mold. Physarum grows in complex networks that are efficient and redundant. They're efficient in that they use a small amount of material to connect the dots. In this case, the dots are sources of food for the Physarum. And it's redundant in that it creates a network where you can remove one of the lines, or one of the lines could be damaged, and the system as a whole, the organism as a whole, can still keep all the dots connected, more or less. So we used this logic, the logic of slime mold networks, to create an algorithm to create potential partition designs where we're connecting the dots of what this partition has to do. It has to connect to the points above and below. It has to hold some equipment. And we used a process of artificial evolution to generate, evaluate, and evolve literally thousands of different design options to explore a wide design space as the process of natural evolution, biological evolution itself does. But in this case, we're doing it in the computer. So here's a different view of our data set. Each point on the graph is a different design option. We can sort the designs according to a kind of color coding, which might be the kind of virtual DNA of the designs, different types of design solutions. We can use algorithms to identify the best set of designs shown here in the black outline. We can graph them according to our goals. In this case, we want to have the lowest possible weight shown on the x-axis and the lowest displacement under structural load shown on the y-axis. We want to be at that 0, 0 point of the graph. And there are a number of different ways to do this. A lot of these ways are slightly unusual and beyond a typical engineering approach. And just like nature and biology itself, we can run this process at a different scale to rerun it. So here I'm showing how, if on the left we have the problem statement, in the center we have kind of a first round of artificial evolution, creating this crisscrossing pattern of bars. Then in turn, we can create every single one of those bars out of hundreds of microlattice bars shown on the right. And to give you a different view of that, instead of using stock material as the bars, we're creating these bars out of microlattice bars, so kind of a smaller scale bar. Each bar is sized according to the amount of stress that it has in it, somewhat like human bone. And like many of our projects, we aim to kind of test some limits of what was possible with the new design approach, but also to make it out in the world. So here you see the 3D printing process. We're making what is one of the world's largest metal 3D printed airplane components. And we're doing this in order to see if this type of approach could create a new airplane component that could fly in today's planes. So here, kind of zooming out, you see the first full-scale component created with this approach. Here's a kind of digital simulation of this component and how it's going to perform under something like the 16G test, or a kind of static test where you're kind of pulling on the component. And if that was the digital test in the simulation, here is the corresponding physical test. This was just a few months ago, and you look closely in the background, you can see some very excited but nervous Airbus engineers as we're testing the performance of this component, pulling on it with the weight of 24 times the force of gravity to see if this new approach, this new partition can hold together. It did, and in the end, we have created a new airplane component that is about 45% lighter than the traditional component. It's even just a little bit stronger. And the bottom line is that if this component is installed in all of the Airbus A320 planes flying today, this could save about 1 million tons of carbon emissions per year. So the second approach is called biosensing. Which I define as, again, using actual living organisms, not the metaphor of biology, but actual living organisms, in this case, to detect and make visible invisible conditions of the environment. In this example project, in collaboration with some amazing artists and engineers, including Natalie Jeremijenko, we have been exploring a public interface to water quality in the East River of New York City. We do this by creating a floating network of lights with sensors below water to detect things like presence of fish and water quality, and dynamic LED lights above water. Initially, we were using this kind of material, you could call it, these kind of components, digital sensors, electronic circuits. But in a new version of the project, we've been using biological sensors, combining biosensors with digital sensors. And by this, I mean we're using actual living mussels, the shellfish. And here you can see a kind of sped up video of the way that mussels open and close their shells a small amount as part of their natural metabolism. They do this to eat and to basically breathe dissolved oxygen underwater. And it turns out that the rate and the amount that mussels open and close their shells is an incredibly sensitive detector of water pollution. So with that in mind, we can take some living mussels, attach to one side of the mussel shell an inexpensive magnet, attached to the other side a Hall effect sensor, which is basically sensing a magnetic field. And for just a couple of dollars, we can have a better tell-all sensor of water pollution and other stress in the water than, say, a $10,000 dissolved oxygen sensor. So this is obviously good for a project budget, but probably more importantly and more profoundly, this suggests a new idea and a new approach, which is to combine artificial intelligence with natural intelligence, to combine artificial intelligence, kind of the best that computation has to offer, the holy grail of AI, with a kind of natural or biological intelligence that has kind of been there all along, that in many cases has evolved over millions of years to perform very complex functions, sensing functions, and other kinds of functions. And we can combine them together to get a kind of multiplier effect. So here's one of the early prototypes of our installation. You can see the lights changing color because there's a tipping point in the water quality. The water quality is slightly improving at that point. This is a kind of application of biosensing that we think, again, could be applied to much more than just water quality sensing, but could be a way to use living organisms out in the environment as part of our design palette as designers and architects. So the third and final approach that I'll show to round out the three approaches is what I call biofabricating. And this is something that I define as using actual living organisms as tiny factories, basically, to manufacture renewable building materials. So in this project, which again had a number of amazing collaborators, we started with a kind of thought about the Earth's natural carbon cycle, the endless loop of growth and decay and regrowth. And we had the kind of hypothesis that maybe we could take low value raw materials, basically take waste instead of plants or instead of high value materials, spend a very small amount of energy to convert those raw materials into building blocks, create a useful building, and then at the end of the life of that building, return all of that stuff, all of that physical matter of architecture back into the carbon cycle. How could we possibly do this? Seems like a kind of bold idea. Well, again, we turn to a living organism, in this case, mycelium, which you probably already know about. It's the root-like structure of mushrooms. It grows in these branching patterns. You can see the nutrients kind of coursing through these filaments of mycelium. It creates these incredible patterns and forms, but we were actually more interested in the function of mycelium than the form, because it turns out you can combine mycelium with agricultural waste, not the high value part of agriculture, such as corn kernels, but corn stover, the low value waste part of agriculture. Combine that with mycelium, put it in a mold of almost any shape, and in just about 5 days, it grows into a solid object. This is a process that's being explored in a number of different ways, including by Ecovative, a company that we worked with on this project. Ecovative is making packaging material out of mycelium. And our idea a couple of years ago was to create an architectural brick out of this material. And because no one had created large-scale outdoor architecture out of this material, we had to do a huge amount of testing. We tested single bricks, compressing a brick to the force of 10,000 pounds, more than it would ever have to resist, compressing small assemblies of bricks. And we did this so that in the end, we could try to create a full-scale structure out of the material as a test of its viability. So here we are in the courtyard of MoMA PS1, creating a structure that will last, or that lasted for the summer, as these projects do in the competition for the summer Young Architects program at PS1. Here you can see us combining into our design ecosystem not only some technical performance and some biomaterials and biofabrication, but the expertise of a number of different people and categories of people. So we combined the expertise of Columbia University graduate students in architecture who know a lot about form and computation, and the expertise of New York City brick masons who know a lot about stacking objects. Neither one alone had ever made something like this before, but together they kind of solved some problems on site to create this experimental structure. Here you can see the structure in the context of some of the rest of New York City. So in the background, you have some of the glass and steel skyscrapers of Manhattan. In the foreground, you can see the red clay bricks of MoMA PS1. And our structure was kind of familiar, but also new, framing the environment. We were experimenting with not only the technical performance of this material, could it be strong enough, could it be weather resistant enough, but also kind of the creative capacity of the material, the atmospheric potential of the material. What would be the qualities of light and shadow on this material? What would it feel like to be inside it? What would it smell like? But of course, the ultimate test of any MoMA PS1 summer installation is its ability to host a good party. So here is the first Saturday of that summer when we installed it. And you can see 5,000 people arriving to hear experimental electronic music. It was a thrilling moment for us to unveil this prototype, but also terrifying as people were walking inside our structure, rubbing it, scratching it, climbing it. And when darkness fell, we breathed a sigh of relief. But it was actually very fitting for us to test this idea, this new biofabrication process, this new material out in society, out in culture, more or less in a kind of public space rather than only on a lab bench indoors or on the fenced off corner of a construction site. The project also kind of engaged a type of dialogue and public reaction through this new kind of short form criticism and discussion. And at the end of the summer, we took the structure apart a little more carefully than you might typically do. We took all of the bricks and crumbled them into smaller pieces. We combined them with yet other living organisms, bacteria and worms in a compost site. And in just about 60 days, all of the physical stuff of that piece of architecture, all of the matter returned to compost, which we in turn used for New York City tree planting and even for community gardens, more or less proving that this is a material that's non-toxic enough to eat. One of the bigger ideas of the project is that maybe as designers and architects, we can think about designing to disappear as much as we normally think about designing to appear. And so if I've kind of tried to paint a picture of 3 different approaches we've been exploring and shown some example projects of the kind of hands-on prototypes we make in each approach, it's important to note that these projects are very open-ended. They're not ends in themselves. In a way, they're never finished. Instead, each project potentially generates new questions and new possibilities. And with some of these questions and possibilities, they can grow into new projects. I want to show one example of that now. And explain how with our project Hi-Fi, which I just showed, the installation for PS1, our bricks were alive during their formation, but then they became dead, inert, and predictable when we dried them out, and they became more or less like other building materials during construction and occupation. But we always had this question, what if the bricks were not dried out, and if the material did not become dead and inert? What if building materials could be literally alive? Would they have some of the same properties of living organisms, like self-healing and self-attaching and growing? And this is an idea and some questions that we wanted to experiment with, and we found the opportunity in a new installation a few months ago at the Pompidou Center in Paris. So we started experimenting with growing new units of material and placing them next to each other when they were still growing and alive. This was our kind of hypothesis, and we quickly saw that we were able to achieve what we call bio-welding. Basically, the living units fuse together, they grow together, and we have, in essence, brick and mortar in the same object. So here in this time lapse over a couple of days, you can get a sense of how it works. These objects that are sitting next to each other grow into each other and create one bigger object. And this gave us many ideas for architectural experiments. I'll show a couple of them here. We first looked at combining bio-welding with the process that we call voxel 3D printing. This basically means stacking small objects, small cubes or spheres, in a way that's programmed to create an object that consists of hundreds or thousands of units. But the key for us here is that we were not just stacking inert objects, we were stacking some living objects and some non-living objects. And the key was that we had 2 different types of units, mycelium units and wood units, which created 3 different types of adjacencies. So we had mycelium to mycelium, which would bio-weld together. We had mycelium next to wood, which would also bio-weld. But we had wood next to wood, which would not bio-weld. And this gave us a way to program the arrangement of different spheres in a way that it would grow into an object of a form that we were interested in. So here you see after we've had the bio-welding occur, had certain objects stick to each other, other objects not stick to each other. We pull out all the ones that don't stick to each other, and we have a new type of form that could be considered a prototype for architecture, with mycelium units on the outside, wood units on the inside, and we get a small scale version of what could potentially be used to create things like shelters. In a next line of research, we were interested in, again, using biowelding, but this time combining it with what's called material jamming. So jamming is a material effect that occurs when units are poured on top of one another. So consider grains of sand as a unit. If they're poured on top of each other and the grains are spherical, it's going to create a pretty flat mound. If the grains are more angular, it'll create a slightly steeper mound. And if the grains have a kind of forked shape, then you could create even steeper mounds and almost like a vertical wall, potentially. Then you can combine jamming with a formwork, and this is something that's been studied in different applications, including funnels. So in something like a grain silo where you have a funnel, there's a lot of research on how to avoid jamming. You don't want the objects to kind of clog together and create a sticking point. But we were interested almost in reversing the idea. What if you deliberately created something like a clog in something like a silo or a different shape of a formwork? Could we create a more or less poured version of architecture through jamming and then biowelding? So you see some tests at different scales in those prototypes in that video. We used those tests to develop a kind of rough idea of a design we were interested in. We couldn't ensure exactly what it was going to look like, but we set up the process and the rules to try to create a new, more or less architecture scale prototype inside the Pompidou Center in Paris. So this will just show a few of the quick steps. Here we're, again, growing mycelium blocks. We're using simple buckets as the formwork. And we're going to grow them in two separate phases. The first phase allows the units to kind of solidify into their shape. So it goes from a loose material of mycelium and agricultural waste into a kind of solid object. But then there's a second phase of growing when the objects are next to each other. And meanwhile, we take those objects and rather than stacking them one by one like typical brick stacking, we pour them into a kind of formwork with very specific dimensions and properties. Here you can see us building the formwork. It's more or less scaling up from what was shown in the previous clip of the prototypes. Here you see the units inside of the formwork. And then we critically remove more or less the plug at the bottom of the funnel. So if you think of that box as a kind of funnel, we remove the bottom. Luckily, there's some jamming, which means the objects aren't just pouring and flowing out of the bottom of the plug. We remove the looser objects, but there is a kind of self-organized form of an arch of the objects that are jammed in more together. This follows the line of force. And then we allow the objects to grow together. So the first process is jamming. That creates a kind of void at the bottom and an arch shape. The second process is biowelding over 5 days. And here you can see then when we remove the entire formwork, we have a somewhat self-organized construction of a physical arch made from jamming and biowelding. Okay, so finally, I want to wrap up and just say if those first three approaches sometimes generate a kind of spin-off project, sometimes just thinking about this domain of biodesign or really any domain in a design ecosystem can lead to kind of new veins of research. And I want to show the last project as a new vein of research that we've explored recently. So this is a project that's still in progress. It involves some ideas about ecosystems, architecture, and public health. It's a collaboration with two good friends and colleagues of mine, Kevin Slavin and Elizabeth Henneth. And it builds off some amazing work that they and others have done at the intersection of biology and cities. So this may be a little bit familiar to you, but we started with the hypothesis that just as we are increasingly aware of the bacteria in our own bodies and the way a gut microbiome contributes to individual health, we might also start paying attention to the bacteria in our cities and the way an urban microbiome contributes to collective health. In other words, microbes are all around us, even though they're invisible. They're in the air, on our food, inside our architecture, on our architectural materials. And contrary to 20th century thinking that microbes equal disease, and therefore we should aim to make our environment sterile, we now know that most microbes, including, I should say, most viruses, actually help keep us alive. So in this context, for an installation at Storefront for Art and Architecture, we set out to explore two things, maps and models. For maps, we were measuring the microbiome at a gallery in Soho and comparing it to the microbiomes of other places in the city. And for models, we were exploring design scenarios for architecture that promotes microbes. And we're thinking about whether there could be something like probiotic architecture, which might improve public health. To do this, we developed a specific bioreceptive material that was kind of designed to catch and host microbes. And more specifically, we started with wood. Wood is pretty well suited to hosting microbial life based on its molecular composition and its micro and macro shapes. And at the Storefront gallery space, we transformed the facade, the panels of the facade, with wood tiles cut from standard lumber and then deliberately eroded through a sandblasting process to create diverse microclimates. Each microclimate had distinct grain and knots of the wood. You can see a few of them here. That would create different pockets of shade and moisture, different kind of conditions for microbes to grow. And this allowed us to see that even some of the most common and humble building materials, like a cheap 4x4 post of Douglas fir, you know, a forgettable piece of lumber at a lumber store, even that object has a material story that could be uncovered and that might offer a range of visible and invisible effects. So in terms of the physical materials, with a building envelope, as you see in the background there, made of a bioreceptive wood, architecture can become a kind of sensor for the urban microbiome. And for the second part of the project, in terms of living materials, we set up basically a small biolab inside the gallery in order to sequence DNA on site to see what was going on. I'll describe this very quickly. I'll be done in 1 minute. So here are the basic steps. First, swab sandblasted tiles from each location to collect the surface microbes. Then place the swab in a liquid solution and agitate it to break down the cell walls. Then separate the DNA from other substances. Add marker beads to the DNA fragments and produce a pure DNA solution. Load the DNA solution onto a flow cell to be processed by a portable DNA sequencer. This can basically just be plugged right into a laptop through the USB port. And the sequencer converts electrical charge from the flow cell into sequence data, the A, T, C, and G of genetic code. So finally, after we sequence the DNA from each location, the gallery in Soho and some other locations around the city, we designed a system to automatically draw the output as an early and very rough map to an uncharted territory, what's going on in the invisible microbial world in our urban environment. So this map is raw and it's at a coarse resolution, but it's already allowed us to see some interesting things. For example, in the Brooklyn Navy Yard, we saw microbial DNA that performs the function of breaking down heavy metals, which basically indicated that the Navy Yard had specific pollutants that were not found in Soho. And so basically what this project has done is allowed us to use a gallery as an urban space that we're converting into an active living experiment, architecture as a living experiment. We're exploring a new type of architectural facade as a new way of seeing some of the important matter and organisms that help keep us alive. We're looking at materials and their layers of living matter, such as invisible microbes. We're reframing buildings as stewards for the urban microbiome. And the initial evidence indicates that our collective future, the future of all humans plus all non-human animals and all microbes, is probably a lot more collective than any of us can see or imagine. Thank you.